going to do Nesseria today, and for the Nesseria's morphology, we have from N gram negative, from N non-motile, non-sporing, aerobic, and it is Diplococcus found in pairs. Now basically two classifications which are pathogenic, N meningitis and N gonorrhea. This Nesseria grows on chocolate agar or blood agar. But mostly and most preferably it is grown on chocolate agar. So now first we'll do Nesseria meningitis in this video. N meningitis. What does N meningitis cause? N meningitis causes meningosemia. This is the condition caused by meningitis. Meningitis enters through respiratory tract through droplets or aerosolar infection. Then it enters into blood. Meningitis basically attacks the CSF of a body, which we can take out or we can investigate through lumbar puncture, and meningitis causes meningosemia. I have told you that we can check it by lumbar puncture. Lumbar puncture. So this was the introduction. Let us go into the details. First, let us talk about the morphology of N meningitis. With the same technique, we'll do from N it is gram negative, non-sporing, non-motile, and non-capsulated. Now next, what is its shape? It is oval in shape. Oval, diplococci, found in pairs, and spherical. Meningosemia is acute fever. It is acute fever and there is uncomfort. Also, metastatic movements of joints, eye, ears and lungs are seen. I'll tell you once again. Meningosemia is a condition of acute fever, malacia, which means uncomfort, and sometimes there is metastatic involvement of ears, eyes, joints and lungs. Meningosemia is characterized by shock and multi-system failure. So from here you can think of that this is very dangerous disease. Meningosemia is characterized by shock and multi-organ failure. What is virulence factor in meningitis? Virulence. So first is it invades immune system, in which it produces IgA proteases and polysaccharide capsule is it does overstimulation of immune system. It releases lipopolysaccharides. It produces lipo-oligosaccharides, not polysaccharides, oligosaccharides, which act similar to lipopolysaccharides, and this causes fever and septic shock. This causes shock and fever. Now, we should also know what other organisms cause meningosemia. First is pneumococci, then H. influenza, and staphylococci and E. coli causes neonatal meningosemia. Now, the last part of any of the microorganisms that we do in microbiology is the lab diagnosis, in which we go by specimen, collection and transport, direct microscopy, culture, biochemical reaction, and then finally treatment. So for specimen, what you can think of that we can take here is CSF, blood. These are two main specimens that we can take. Others are petechial lesions, and swab from nasopharynx. Swab from nasopharynx is important for detecting the carriers of the disease. Collection and transport. Collection is done by lumbar puncture, I told you, and vein puncture for the blood. Now then we always do direct microscopy, but one thing I forgot to tell is that CSF sample is never refrigerated. Now in direct microscopy, if we have taken CSF sample, then we do three parts of it. First part of this is incubated overnight. One part is centrifuged. Centrifuged and smear is made for gram staining. One is done direct culture. Now in gram staining we know the result, that is gram negative, so pink color diplococci will be seen. Then comes the culture. CSF is cultured on blood or chocolate agar. 35 to 36 degrees Celsius with 5 to 10% of CO2. Now this is very important because in the blood culture also, when we use the blood as specimen, we do it on blood agar and chocolate agar with 35 to 36 degrees Celsius temperature and 5 to 10% of CO2. The results will come after 28 to 24 hours in CSF, and in blood it will come after 24 hours. For the blood, either we can use blood culture bottle which will have glucose broth and the temperature remains the same, and we'll get the result in 24 hours. For others also, blood and chocolate agar with the same conditions, it will be done. Now, how will colony appear? Colony will be round, convex, grey, translucent with clear edges, and diplococci. Biochemical reaction, it is catalase positive, oxidase positive, and maltose will be fermented acid and not with gas. Slide agglutination test can be done. Serological test can be done for chronic meningococcal septicemia, and PCR can be done to increase the DNA of meningitis, and then seen further. Treatment is, these are sensitive to penicillin. If a person is resistant to penicillin, then chloroamphenicol can be given. Thank you.